Hey everybody, this is Bima and you are watching Zufo. Please press the subscribe button and bell icon right next to it. Okay, in the last class we have studied about the microsporogenesis and now we will move on with the megasporogenesis. Before that, in the last video that I have forgotten a bit about the microsporogenesis. It means in the pollen structure, where I have said that pollen is composed or it is covered by sporulanin. But there one question arises. If that strong layer is present on the pollen grain structure, how come the male gametes come out? Yes, I have forgot to tell this. So I am telling it now. So there bit structures on the on the enzyme, there a bit space is present, pore-like. Those are called as the germ pore, where the sporulanin substance is not present there. So through the germ pore, male gametes comes out, and the coming or further next process will goes on. So now let's move on with that female reproductive part, that is gynoecium. Gynoecium is the female reproductive part where it may consist of a single pistil or many pistils. So if it consists of a single pistil, then we call it as a monocarpellary. If it has a more than a pistil, 2, 3, 4 pistils, we call them as the multicarpellary. So in pistil we find three parts, that is the upper stigma part, where stigma acts as the platform for the pollen to takes place, and the style is the elongated structure, and the bulged or the basal part, that is ovary, where ovule or oval cavity locules are present in it. So here these are the pistils or the carpellaries are in the two forms, that is they may occur in the fused form, where more than a single or more than a pistil, 2 to 3 pistils, if they are present, they are called as the syncarpus because they are fused. If at all those pistils are not fused, we call them as the apocarpus. Please remember these words, syncarpus and apocarpus. In syncarpus, these pistils are fused, and in apocarpus, these are not fused. So this is the small brief explanation about the pistil, and please remember this monocarpellary and multicarpellary. It's important for your exams also. So let's go on with the structure of the ovule, where this ovule structure or the ovary is present on the placental surface. So here a stalk like a structure which is, with that help, this ovule is present on the placenta. That stalk is called as the funicle. It is a stalk like structure which is called as the funicle. And next comes one more hilum, where this is the point of the attachment of the funicle with the body of the ovule. So this part is fused or it is attached with the body of the ovule. This is called as the hilum, and a bulge structures occurs in the or below the funicle, that is called as the raphe. And these ovary or these ovules are protected by one or two envelopes or the layers called as the integuments, which are protective in nature. And these ovule possess two ends, that is basal and chalazal. We call it as a chalazal end, that is the basal end. And the one more is, that is upper end, where it is called as the micropylar end, and it will be having a bit opening. It will be having a space for the entry of the male gametes. That is, so two ends are present, that is chalazal and micropylar end. And inside that numerous cells are present. Mass like a structure is present. That is called as the nucellus. In nucellus, that embryo sac is present. This nucellus reserves large number of food materials, means it will be possessing reserved food materials, and it is composed of parenchymatous tissues. Inside the embryo sac is present. Before that, the MMCs, that is megaspore mother cells, before the formation of embryo sac, These MMC, megaspore mother cells, undergoes meiosis division as in the microsporogenesis occurs. Here also the meiosis division occurs, and this MMC gives rise to 4 tetrad megaspores. In these 4 tetrad megaspores, 3 megaspores gets degenerate and one cell, that is 1 megaspore, will be functional in order to form the embryo sac. This one remained functional megaspore cell, it undergoes mitosis division. 3 times it undergoes mitosis nuclear division, where a nuclear, in the first division it undergoes nuclear division and gives 2, later 4 nucleate, and later it gives 8 nucleate stage. So this is how the 8 nucleates are formed by the division of the nucleus. Here 3 mitosis division occurs. Please do remember it. After that, these 8 nucleate are formed, and then occurs the cell differentiation, so that forms the embryo sac, in which 3 cells goes and sits to the chalazal end, and remaining 3 cells goes to the one more end, that is micropellar, and remaining 2 nuclei that sets in the center, that we call it as a 2 polar nucleus, and that whole cell is called as the biggest central cell. So the upper chalazal cells which are formed and sat, those are called as the antipodal cells, and the 3 cells that is called as the egg apparatus, which includes 2 synergids and 1 egg. These 2 synergids and 1 egg, that is called as the egg apparatus. So here are the 8 nucleate, that is 3 antipodal cells where 3 nuclei are present, and in the micropellar end 3 nuclei are present, that is 2 synergids and 1 egg, and in the middle 2 polar nuclei are present. This is 8 nucleus stage. And how the 7 side occurs? Because upper 3, that is antipodal cells, and here egg apparatus consists of 3 cells, and coming to the central, that is a central cell, we should consider it as a single cell. So here comes 8 nuclei, it is 8 nucleate and 7 cell stage. They may ask you this question as it is very important. Please do remember it. Here in the synergids, there are structures which are filiform like structures, which help in the, or which they guide the pollen tube to attract towards the ovule in order to the occur, or they helps in the fertilization. This is the small information about the megasporogenesis. Please do remember it. And in the next video, let us go through the one more topic, that is pollination. Till then, have a good day and bye bye. Take care.